Okay, now it's working. Sorry. Can I turn off my camera for better connection, or
Yeah, sure,
it's
sure.
necessary to?
Try that. That will be very useful.
Okay. Well, hello everyone. Sorry for my delay. I'm going to present some of my results obtained during my postdoc position here in Córdoba. I am working at the same place of Rocio, and my work is about the diversity in the repetitive DNA fraction in Solanum betaceum, Solanaceae families. First of all, I want to talk about the evolution of genome size. We know that within the plants, we find that there are some of them with very small genome sizes, like Genlycia tuberosa, with 0.06 picogram of DNA. And in the other extreme, we have plants with giant genomes, such as Paris japonica, with 152 picograms of DNA. Despite this great difference in the genome size of the plant, most of them are have small to medium sizes genomes. The possible causes that decrease the genome sizes are an unequal crossing over, higher rates of deletion than insertions, and selection against transposable elements. Among the causes that increase the genome size, we have polyploidization and transposon amplifications. In this graph, we can see the sizes of the main gene families has increased their sizes throughout the evolutionary history of the plants. And this is mainly due to the polyploidization and accumulation of repetitive sequences. But what are these repetitive sequences and how they are classified? We know that in each genome, we have single copy sequences like genes and repetitive sequences. These repetitive sequences can be tandemly repetitive DNA sequences. That means that they are distributed next to each other in a head-to-tail type arrangement. These are called satellites or ribosomal DNA. And in the dispersed repetitive sequences, we found the transposon and retrotransposons. The retrotransposons are classified in type 1 or type 2, according to the mechanisms that they use to move in the genomes. Type 1 are those that copy and paste elsewhere in the genomes, and type 2 are those that cut and paste elsewhere in the genome. The first one are the more responsible for the increase in the genome sizes. Here we have a graph. We can better see what is a tandem repeat sequences, each in the head-to-tail arrangement. For example, satellite or ribosomal DNA is here, or here is another example. And here we have the sequences scattered or dispersed in the genomes. These retrotransposon sequences are classified according to the protein domains that they have or they possess and the order of these protein domains. Well, the Solanaceae family is a medium-sized cosmopolitan family, as you know, with 99 genera and about 2,700 species. This family has representative of great economic importance, such as eggplant, tomato, and potatoes. Within the Solanaceae family, the genus Solanum is one of the most important in terms of numbers of the species. And regarding the evolution of genomic sizes within this family, we see that the majority have small genomes, like we see in red here, in this graph. But there are some genera with medium sizes genomes, in green, for example, capsicum or solanum or cestrum. And genera are some genera or genus, sorry, such as cestrum or cesea that have giant genomes, like here in blue. But within the Solanum species, there is a clade or a group of species that has giant genomes. They are known as Siphomandra clade and are the wild relatives of the tomatoes. So in detail, we can see that this Siphomandra phylogeny is made out of different subclades. So here we decide to start studying the first clade that is Solanum betaceum. Solanum betaceum is native of South American, and it's known like tamarillo or tree tomato. This species grow in several countries for commercial use, for shoes, sweets, or nature consumption. Sorry. So our objective in this work was to characterize the repetitive fraction in the genome of the tree tomatoes and other related species, all of these with giant genomes. For this, we use the methodology. We prepare the DNA extractions for Illumina sequences, and the genome size were estimated by flow cytometry. Bioinformatic analysis were performed in the RepeatExplorer pipeline. And then we prepare proofs for quantification using dot plot and for fish experiments. The repetitive fraction in Solanum betaceum represents 76% of the genomes. The most abundant element is a gypsy tecai, followed by an Attila. All of them are retrotransposons. And we can see that the portion that represent DNA satellite is also important in the Solanum betaceum genomes. We found 7 different satellites family in the genomes. The first satellite we called satellite 1 because it's the most abundant satellite in the genomes, and it's a medium conserved satellite with 299 base pair. This satellite is co-localized with the CMA plus band in the terminal and pericentromeric regions of some chromosomes, not all the chromosomes. The second satellite is called satellite 2 and is 90 base pair satellite, and this satellite is more conserved than the previous that I showed. This satellite is located in the terminal region of most of the chromosomes. Here we can see that it's a small dots distribution of the satellite that is colocalized with this big CMA plus band. And the satellite 3 is a large satellite with more than 3,000 base pair. And in the fish, we see that this dispersed throughout the genomes. And for the next 3 satellites, we didn't obtain signals on the fish experiments. So the next one that we prepared the groups was a satellite 4. It's a satellite with 1,500 base pair. And show us a dispersed pattern in the genome as well. So after this result, we decided to compare 5 different populations from Argentina, from different geographical areas, because we observe a phenotypic difference between them, such as fruit color, sizes, etc. First of all, we compare the genome sizes of this population, and we saw that some population present a difference in genome sizes, which vary from 22.5 to 23.5 picogram of DNA. In order to compare the different population, we did a comparative analysis of the repetitive fractions using the same coverage for all populations. So in this graph, we can see that each color represent a different elements, and the sizes of each bar represent the abundance of each element. So you can probably think that all looks similar. If we observe, for example, some cluster of retro element, this is the supercluster 1, is the more abundant supercluster in the genome, and this is all the cluster that form this supercluster 1 is a TK element. And we can see that the abundance of all population, or this element in all population, is similar. But if we look at the relative abundance of this satellite DNA cluster, for example, here in orange are the satellite DNA, we can see that they vary more than the abundance of the retro. This is one satellite, for example, and this is the other satellite. You can see that here is more abundant than here. This is the other satellite, and this is the last one. You can see that in this population, this satellite is very, very abundant, and not in this population, for example. So here in detail, the graph of this satellite, with each color represent each populations. This is the first satellite we described, and here we can see the different population and the relative abundance of these satellites. And we can see that the abundance is different in the populations, different populations. Here we have the satellites too. The same is very, very different, the relative abundance on the populations. Here we have the satellite 3, the 4th satellites, and we observe difference in the 5S and in the 45S. And what we know about the other species? Now we are working with species from these two subclades within the Siphomander clade. And the first, Solanum glaucophyllum, has a genome size of 12.5 picograms. Solanum confusum, 21.7 picograms of DNA, and Solanum stuckerti, 17.3 picograms of DNA. Regarding the repetitive fraction, this represents 54% of the genome in Solanum glaucophyllum, 63 in Solanum confusum, and 53 in Solanum stuckerti. In these two species that are more related, here confusum and here stuckerti, both of them have the more abundant cluster is the same, is a decay element, but in Solanum glaucophyllum we observe that the more abundant cluster or retroelements is an ogre, is different. For Solanum stuckerti, we describe 4 different satellites. For Solanum confusum, 4 different satellites too, one more abundant than the others. And for Solanum glaucophyllum, we describe 9 different satellites. So we can see that there is a difference between these species. And in the comparative analysis, in this case, we can observe an interspecific variation. For example, we observe that there are satellites here in pink that are conserved and shared by the 4 species that we are studying here with different abundance, but it's the same sequences. Other satellites are species specific. For example, here one satellite is only present in this species genomes, and other satellites, for example, is present and in Solanum stuckerti and Solanum confusum that are more related species, more closer species. So for concluding, we have in this work, we have observed that there retroelements responsible for the increase in genome size are decay, followed by Attila, except in Solanum glaucophyllum, that is an ogre elements. In Solanum betaceum presents a greater accumulations of repetitive DNA compared to the other species analyzed here, and the genomic sizes of the population vary as well as the relative abundance of satellite DNAs. So thank you for the invitations.
Thank you, Mariela. That was a great talk and a lot of results about this repetitive DNA in these different species of Solanum. So now we are open for questions. Lynn, want to?
Yes, I have some questions. Nice to meet you, Mariela, and this is Lynn Bose. As you know, I'm very, very interested in this clade.
Yes.
My first question is about the sampling within Solanum betaceum. It looked like from your map, I couldn't tell exactly, it looked like you might be sampling some of the populations that we think represent the wild populations of Solanum betaceum in northwestern Argentina. Is that where they're from?
They are from Salta, Tucumán. One of the sample is I took from the databases, so I only have the genome the sequences, not the plants. But
I see. Okay. So I'm interested to know if this type of investigation into genome size could help us to say something about the history of the cultivated tree tomato.
Yes.
So, you know, in northwestern Argentina and southern Bolivia, we think that those populations are truly wild, and that's the original home of Solanum betaceum. But it's actually not very widely used in Argentina. If you go to the northern Andes, to Peru and Ecuador, there's tree tomato, tomate de árbol everywhere. And so I've been interested to know how we would sort out the movement of that species throughout Latin America. And I'm just wondering if your type of research could help us to sort that out.
Yes, I wanted to sequences other species, not only tree tomato, and maybe prepare a phylogeny analysis with the repetitive sequences to compare with the traditional phylogeny.
Okay, that would be very interesting. And my suggestion is maybe to think about sampling within Solanum betaceum, but from, for instance, the northern Andes, from Mexico, from other places where it's been obviously dispersed somehow, sometime.
Yes, yes, it's true. It's a good idea.
My second
Thank
question is, it's very strange that this clade is distinctive in having these giant genome sizes, including the Cyphomandropsis species that are in that clade, and you sampled them, Colchophyllum and Confusum and Stackertii. So that appears to be a synapomorphy of this clade within Solanum, and I don't know of any other clade within Solanum that has these huge.
No.
Do you? No.
No.
But the other characteristic that is distinctive for this clade are the anther-connectives and their odors. And so I have no idea, but is there some effect on phenotype of having these repetitive elements in your genome? Or are these just kind of characters that show us the direction of evolution, but they're unrelated to each other?
Yes, it's very interesting that because the difference in the genome size is very, very absurd, because the tomato genome sizes is 1 picogram, and in the tree tomato is 23 picogram. Is very, very different. So we wanted to characterize sorry, it's very difficult, this word, these species, and now we are thinking in another project in collaboration with Germany to try to know why some species of plants can accumulate a great quantity of retro. Why is the mechanisms why they can accumulate, and other plants, another species, related species, they cannot. So this is a future idea for me.
That's very interesting. Thank you. I'll be following your work because I really love these plants, and they're
thank you.
Super interesting. Thanks.
Thank you.
Thanks, Lynn, for your question. I don't know if there is any other questions there. Any other question or comment? This whole war about repetitive DNA is so intriguing, and there are so many questions all the time about the evolution of that particular type and how expanded in some species and not others. That's really cool.
Yes. Yes.
So Ellen is saying, thank you for such a clear talk. I learned a lot.
Thank you.
Okay.
Thanks, Mariela. That was very, very interesting.
Okay, thank you.
Great.
Worth getting up at 7 in the morning for.
Yeah, thank you, Vic, for doing such an effort. That's right.
Where are you from?
Seattle, Washington. So we're maybe 4 time zones different from you.
Thank you.
Yeah. Okay, so if there is no other question, I'm saying goodbye. So this was the last seminar of the year. So we are meeting again next year. And I hope to see you all in Madrid in the International Botanical Conference, where it will be there is a symposium Solanaceae, many talks there as well. I hope you submitted an abstract there. I still have to check them, but yeah. So, well, thank you, everyone, and see you next year. Happy holidays.
Thanks. Same to you.
Thank you. Bye, everybody.
Thank you. See you.
Bye.